Hello, everyone. My name is John Severin. You made it to the last session of the day, and we'll be focusing on CMT1A during this last session. I'm a faculty member here at University of Wisconsin. My lab studies CMT1A and things around CMT1A. I also serve as chair of the Scientific Advisory Board for the CMT Association, and I also am a member of the board as well. What I'd like to do in about 10 minutes or so is just sort of recap some of the highlights, some of which have been covered to some extent in previous sessions this afternoon, but also just to set the stage a little bit. And ultimately, we want to make sure we leave time for your questions. So hopefully this won't take too long. But I just wanted to kind of highlight that, we are really attacking CMT1A from all sides here. One of the sessions that we had was on genetic therapies for CMT using either ASOs or gene therapies or RNA as well. And this is really a highlight, I think, in those efforts. We also have been very active in drug development for CMT, and I'll kind of share the latest updates on things that we know. One of the things that we have really wanted to highlight here is the fact that as we have clinical trials for CMT1A, there's been an immense amount of work to try to make sure that those clinical trials are designed well and they can be done efficiently. And that allows companies to be able to invest in developing new therapies for CMT. And we've worked closely with different companies who have developed their own compounds or their own therapies, and we try to facilitate as much as possible their entry into CMT and lower the hurdles for them to be able to get into clinical trials for CMT. So just a few words, I think, for CMT1A, I just want to kind of set the stage in terms of what the language I use so that you are aware of the terms that I'm going to be using. CMT1A is really affecting the myelin that surrounds axons in the peripheral nervous system. And we talk about it as insulation, but it's more than insulation. It actually makes nerve transmission much faster. And one of the things that I like to say is that myelin really makes nerves go from 3G to 4G. They really accelerate the transmission of nerve impulses to your muscles. And so it's not only the propagation of those nerve impulses that's really important, but also the myelin that's there. And CMT1A is referred to as demyelinating CMT. So the original problem is really the myelin itself, which is formed by Schwann cells. And other forms of CMT, for example, CMT2, are considered axonal CMT. And that means the issue is a little bit more with the nerves themselves rather than the insulation around those nerves. So when we're considering CMT1A, then we really have to really focus on the Schwann cells that are making that myelin and see if there's a way that we can restore the balance in CMT1A in order to essentially address the cause of CMT1A. And CMT1A, as many of you are aware, is actually caused by a gene duplication. This is something that is present in the population. There's roughly maybe 50 to 100,000 people in the United States that have CMT1A and more in Europe and across the world. And there's a small segment on chromosome 17, which gets duplicated. And within that duplication is the gene known as PMP22. And work of many scientists has really indicated that the duplication of this gene, so that we have not just two copies, one from mom and one from dad, but now we have three. The duplication of this gene, PMP22, is really what causes CMT1A. And so in a very simple diagram here, the issue that occurs then is that the output of that gene is higher in CMT1A in terms of the protein that's produced from that gene. And in our lab and many other labs, there's been a lot of study to try to indicate, is there a way to lower that excess protein? And so simply put here is if we can sort of just dial down the gene, we could really solve CMT1A, and we can use genetic therapies and other ways to do that. And essentially, that's one of the things that we're trying to do is find the Achilles heel of this gene, and in order to be able to reduce that level back to normal and bring things back into balance. So the other thing that has emerged as something that's important to know is that PMP22 actually kind of passes through the cell in order to get out to myelin, and there are ports within the cell that transport PMP22 protein out to myelin. And the best analogy I can think of right now is that the ports are essentially kind of gateways that transport PMP22 like that. And all of us are aware is that we've had transport issues in our infrastructure in the United States that's caused stress to the system. And so one of the other strategies in order to try to, ease the problem is try to ease the stress that's caused when you have too much protein trying to get through one of these ports within the cell. So those are, other issues that we can attack in order to develop new therapies for CMT. Now, we had a whole session on genetic therapies for CMT. There was an excellent presentation by Dr. Cleopa. And many of the things that we're trying to do have already been mapped out for us. There's a disease known as spinal muscular atrophy, which was actually quite a devastating disease that, was really life-threatening for many kids. And now we're in the happy situation that there is a gene replacement therapy, there's an ASO therapy, and these therapies were developed and have really established themselves as very, great therapies for this disease. And this disease actually affects the same nerves that actually go out that are also affected in CMT. So the great news is that we can really follow the same game plan for CMT as has been established for these other types of gene therapies. Of course, there's some differences that we have to bring into consideration, but many of these efforts are ongoing as outlined in that gene therapy session. So I want to just reinforce that there's many types of gene therapies. There's AAV therapy, sort of a virus-based therapy. There are antisense oligonucleotides and also RNA that can be used to reduce PMP22. It's very exciting that we've had a company known as Toolgen, which was in a previous session, which is using this CRISPR-Cas9 technology and gene editing. And we also have other groups funded by the CMTA that are looking at this sort of technology. And all of these are really focused on reducing this level of PMP22, as I mentioned. Now, one of the things that's unusual that we have to solve is making sure that we can deliver all of these great types of therapies to Schwann cells. They don't get delivered to the neurons, but remember, the Schwann cells are the issue, and delivering those to Schwann cells is still an issue that we haven't completely solved with. There are many things ongoing which have a lot of potential, but at this point, there's nothing in the clinic that's allowed us to be able to solve this delivery issue. One of the things I want to reinforce is that even though there are basically gene therapies that are in process, we're also still doing a lot of work with drugs or small molecules for CMT1A. This has involved drug testing with the resources that have been developed by the CMT Association. One of the things that's been exciting is there are certain companies that have entered into the space. There's a company known as CKD Pharma, and this is a company that published a paper with some very nice preclinical work in CMT1A, and they have a compound really in phase 1 trials already, and we're hoping that moves to phase 2 trials. There are a variety of different efforts within the CMTA, our own laboratory, testing drugs that reduce the levels of PMP22. We also have other drug targets that are being spearheaded by the lab of Laura Feltri and Larry Rabbits at the University of Buffalo. And there's some exciting results that have been generated in that project. And then finally, there's another compound that's been in development for a while now. It's called from Inflectus. This compound is called IFB-088. And it's also shown some promise in CMT1A models and CMT1B. And phase 1 clinical trials have finished, and they're in phase 2 trials, or they're planning the phase 2 trial as well. So all of these efforts together, again, are complementing all of our gene therapy efforts to try to find really the best kind of therapy for CMT. The last thing I want to sort of cover in terms of activities is just the amount of work that's really required to really try to develop a good clinical trial design. And this came up in that session as well, is this challenge in terms of CMT, that it's a slowly progressive disease. And that means a positive response to therapy may take a while to, be apparent. And the longer the clinical trial, the more expensive it is, and the less likely a company will invest with that. I did put this picture in here because this is the side of an apartment building, and if you want to know how things are going in terms of electricity or gas usage, you can use these meters on the outside of the building to know how much energy is happening. And we need something like this, meters that kind of tell us what the neuropathy status is within an individual, that will be rapidly responsive to treatments. And so we've done a lot of work within the CMTA and in partnership with the Inherited Neuropathy Consortium to develop new sort of biomarkers that can be a readout of effective trial design. And this has included development of the calf muscle MRI. This is a way of assessing the muscle status in the calf muscle, and it turns out to be a very sensitive measure. And we're also working on other kinds of markers that we can use in a treatment regimen, so we can assess whether the treatment is working even at early stages of the clinical trial. And this is immensely helpful to companies that need to kind of know if the approach is working before they invest a lot of time and money into longer trials as well. So some of these things, as I mentioned, are MRI of calf muscle. There's been a lot of work in terms of neurological assessments. David Herman presented in one of the previous sessions, and they've been very active in developing other kinds of sensors or ways to collect data on grip strength and other things as well. Gait as well, as a way of sort of assessing the status of someone with CMT1A, plus also seeing how they might respond in a clinical trial. So all these things put together, can be quite important. And we've been actually quite excited is that there are ongoing clinical trials that are using this MRI of calf muscle. And this would be critical because this would allow us to argue with the FDA that they could use this as a primary measure in the clinical trial, and that would make that trial, less costly and could involve a smaller number of patients and make the trial design much more efficient. So again, I just want to give credit to the fact that the Inherited Neuropathy Consortium is really a worldwide network of neurologists across the globe, and it's taken an immense amount of effort to develop these kinds of data. And I just want to highlight that this network has been really instrumental in providing data to support clinical trials for CMT1A. So last thing I wanted to just recap then is just we've really tried to do a multi-pronged approach to development of treatments for CMT1A. Many of these things are on the cusp in terms of development of preclinical data, and we're hoping that these will translate into clinical trials here soon. The timeline is never fast enough for us, but we have to do due diligence along the way to make sure we have good clinical trial design, and also the ways that make sure that those treatments are also safe as well. So those are very important activities. So with that, those were the major points I wanted to cover, and I wanted to make sure that we had time for questions. So I'm going to stop presenting here. And I think Laurel was going to join me to help answer any questions. Laurel, are you there? There you are.
Hi, John. Okay, so the first question, let's see. I thought I put it on stage, but let me just move this. Okay, so Catherine said, my 33-year-old son has won a more severely with foot drop than my daughter and me. Should we come from Edmonton, Alberta, Canada to the US, or is there no benefit to that? And why is his more severe? And then she also said that 80% of her cousins have it. Why so high? I'm thinking that part's bad luck, considering it's a 50-50 inheritance.
Yeah, that's really an excellent question. So it is a sort of a common thing that sometimes in subsequent generations, there seems to be sometimes more severity of the neuropathy in terms of symptoms. And I think as scientists, we haven't really, answered the question of how that is. It could could be due to sort of the genetics of it. So we know that CMT1A is caused by PMP22, but other things in the genetic makeup of individuals can make that severity, worse or make it more mild. And so, that's a subject that's under study. This is led by Stefan Zuckner in Miami. In terms of traveling to the US, I think having a genetic diagnosis of CMT1A is quite important. I will confess my ignorance here. Obviously, the FDA, governs clinical trials within the US, and I'm not sure how they work with Canadian law with that. I think there may be some eligibility for Canadian citizens to participate in clinical trials once those are announced. And I don't have the answer to whether that can happen or not. But, I think as you see clinical trials being announced through the CMTA and other sources, I think you can certainly inquire about that.
They're actually the PXT 3003 trial that Pharnext is doing for 1A. There are a couple of sites in Canada. The trial is now closed. Enrollment is closed, but it's important to keep track of that. Just to piggyback on what John has said, you might not necessarily have to come to the US depending on what those regulations look like. And there's certainly clinical trials all over the world. So it's just really important to stay tuned and to make sure you're signed up for eNews. I am going to do something different. So Roy Belke has had his hand up all day long. And so, Roy, since you're not typing your question in chat, I'm going to try to hand you the mic. And if you unmute yourself, you can ask your question. Roy, if you can hear me, we'll give you a minute to unmute yourself.
Yeah. Okay. Thank you. I'm wondering whether any of the researchers have looked at the drugs that are being used for MS to remyelinate. Ampera, I guess, is one of them, and it seems to be successful in remyelination. And does this apply to CMT?
Yeah, that's an excellent question. So let me just say that there are some important differences of CMT with multiple sclerosis. And one of the things about MS is that it's really driven a lot by immune reactions that, destroy the myelin within the brain. The cells that make myelin in the brain are different in many respects to Schwann cells. However, there has been some work published by Rudolph Martini looking at different immune reactions that could be targeted in the context of CMT. The other thing that I would mention is that this company known as Inflectus, that drug that they've developed has actually been tested in models of MS as well as CMT, and it looks like it would have promise for both multiple sclerosis and CMT. So there has been a related drug that's been tested in the clinical trial for MS. It had some, side effects, so they would probably have to redo it with the drug that I mentioned as well. But anyway, I think there are certainly some crossovers where you could have something like that. But the immune system plays a really important role in MS, and it's not quite the same in CMT. Great question, though.
Well,
Thanks,
thank you. Thanks, Laurel. Thanks, John.
Thank you. And just a follow-up question to that. I know with MS, it's autoimmune and we're a genetic disease, but is one of the biggest challenges in 1A, John, getting the treatment all the way to the Schwann cells?
Right. Yeah. So I think what I was trying to say is that the delivery of, say, genetic therapies to Schwann cells is quite important. The other thing that we have to deal with that Dr. Cleopa mentioned is that there's a blood nerve barrier. So that means that drugs that are injected, they don't automatically go into the brain because there's a blood-brain barrier, and they don't automatically go into nerves where the Schwann cells are. So drugs or therapies have to be compatible to try to cross those barriers, the blood-nerve barrier. And I think one of the things that's still a hurdle for the genetic therapies is really delivering, CRISPR-Cas9 or ASOs into Schwann cells. There have been some successes yet, but I think most of us think it has to be improved before we actually, really have a very effective therapy as well. And so those Schwann cells are hiding out in the nerve, and so we have to figure out a way to deliver that. There's been various solutions that have been proposed, and that is really an important issue.
Yeah, we just need that tiny detail, right?
Yeah.
So another question just came in. What do you think, Dr. Savarin, is the most promising trial to reverse some of the effects of the disease?
Right. So that's a very important question that is always brought up in terms of reversing the symptoms. And this is somewhat dependent on the level of progression. So, someone who's had CMT for a long time, there is some issues that some of those nerve connections to muscles, they may actually have been lost to a certain extent over time. And so in that case, we're trying to stabilize the remaining connections that exist in order to be able to achieve some sort of recovery. We have some data from mice and rats that, theoretically, it may be that a successful treatment would just stabilize the neuropathy at the current status and not actually, allow things to improve. However, in some of the rodent studies that have been done, we actually not have just seen stabilization, but actually improvement. And I think one of the things that is kind of important is that, the people with CMT, exercise is beneficial, but people with CMT don't get quite as much benefit from exercise as other people, partly because they have this nerve issue. And so we're hoping that if we can stabilize those nerves, then people with CMT would actually then benefit more from exercise and help to, recover some of that function as well. So I think in the long run, my guess is that successful treatment of CMT will probably involve some encouragement to, do certain exercises that may help to recover some function as well.
Wonderful. Okay. I'm a big believer in exercise. I know as a person living with 1A that my body feels better when it is in motion. That's for sure. Couple questions coming in about the FARNext trial. Is there an estimate when it will be available? I know that
Right.
David spoke to that.
Yeah, the FARNext representative was in the previous session, and as I understand it, they're repeating their phase 3 clinical trial. and the data are not available yet. I think if they develop successful data, for that, then it could become approved. I think the information I have is mostly, from the press releases that they have. And so there will be essentially based on the trial that's ongoing, it will have to become, approved by the FDA before it can move into that. So, yeah. Did that answer the question?
I think so, and I think that Dr. Solomon said that it will be at least until the end of fourth quarter 2023, because they have to close the trial, and then I think they'll share as they're able to as they learn. So
Yep.
very exciting. So I don't know, John, if you can speak to this, but we try to help so much with patient recruitment at the CMTA, and the patients who are available to be in clinical trials and studies are the older patients because they're retired, or they're more interested in raising their hand because their CMT is more progressed, but they don't qualify to be in clinical trials because they're older. I'm not sure if you can speak to that at all, the age thing, and why younger people are needed in clinical trials.
Yeah, well, I think, at this point, there's still going to be a spectrum of ages that will participate in clinical trials, as I understand. One of the things that Dr. Cleopa mentioned is that, there's some expectation that it would not be pediatric clinical trials at the early stage. That might vary from trial to trial. And so I do anticipate there'll be sort of a spectrum of individuals, up to certain ages that would be do that. In terms of defining those criteria, that will be sort of an ongoing dialogue between, the neurologists and also companies who are trying to design the most effective trial. I think if you think of it as a standpoint from the company, you would like to design that clinical trial to be have the greatest chance for success. So I think people that have progressed, maybe to an extreme degree, may not be part of early clinical trials, but I hope that they would be able to participate as clinical trials progress. But again, it depends on, the type of therapy, the company that would be designing that, and in consultation with neurologists that know CMT. So anyway, I wouldn't say right now that there would be exclusion of people at higher ages. So I'm in my 50s as well, so I have to put myself in that bucket as well. But I can't really forecast necessarily what age range would be in there.
Yeah. Gotcha. And then someone asked, and I just want to let everyone know in the chat, if you're asking a personal medical question, I am going to skip over your question, especially as it relates to prescription drugs, but you're welcome to send me an email. And then, John, you mentioned the work that you're doing with mice. And the mice, everything happening in the laboratory, the mice models showing improvement. Could you speak a little bit more to that research?
Right. So in the field of CMT, one of the things that's very important is to have a good mouse model of the disease. And we can use either mouse or rat models of CMT1A that are available. One of the models that's quite popular right now is the C3. It's called the C3 model of CMT1A. This has been used in the preclinical testing program that Mark Scheidler mentioned in the previous session. This same model is in my lab. It's in several different labs that work with the CMTA. For example, Dr. Cleopa has a similar model as well. And this is a model that has the human PMP22 gene, and it has things that you can measure that reflect a neuropathy. And so you can use that model to see if you can prevent symptoms from arising, or you can let the model get a little bit older where the neuropathy is more apparent, and then you can see if a candidate treatment will either stabilize the neuropathy or help to resolve the symptoms as well. So this has been a really critical model across the board many of the previous people that have presented, if they talked about using preclinical studies with mice, they probably used the C3 model or something. There's another related version of that. So this is a good way for us to assess that. There is also a rat model that's also used, and that's also within the preclinical testing service that's been developed by the CMT Association. So I think these have been very key things that are developed. Now, for many of these therapies, there has to also be some sort of safety testing.
Mhm.
And sometimes that's done in other species in addition to mice or rats. And that really depends on the type of therapy or type of drug that's being developed as well.
Really exciting. It's truly remarkable. I'm going to grab one last question here. It says, my daughter with 1A has periodic episodes of acute pain in her extremities. Do any of the remedies, probably I think he means research being discussed, address the symptom of pain?
Yeah, so that's a really good question. I know for many people with CMT1A, there is issues of pain. It's been a little bit variable in terms of availability. I think one of the things that kind of comes up when you consider clinical trials is you probably want to use symptoms that are probably most prevalent throughout the population of people with CMT1A to see if you can resolve that. That being said, I think that will also come into account that if someone has painful episodes or pain associated with CMT1A, that'll be certainly something that'll be tracked within clinical trials to see if there's any improvement with that. I do recommend that if you have a neurologist that you see with CMT1A, that you consult with a neurologist in terms of if there's a good way to sort of address the current symptoms. And there may be a strategy that works well, but that it probably is a case by case basis, and it's hard to provide a overall recommendation.
Yeah, I think a lot of that is trial and error. So don't give up if you do have severe neuropathic pain. If your neurologist won't help you, and I hope they do, but if they won't, I would look for another neurologist or a CMT specialist at one of the centers of excellence, or also a pain management clinic or specialist, because they're, with some trial and error, you should get some relief there. So I'm going to start wrapping us up. I just wanted to say that if we didn't address your question in the chat or Q&A, please feel free to reach out to me. Or to our info@cmtausa.org email address, and we will do our best to get all the questions that have come in today. I want to thank Dr. John Svarn for being with us, because he shares his time and his brilliant mind so generously throughout the CMTA and research and everything that we do here, and we couldn't do it without you. So thank you so much for sharing your time. And thank you to all of us for being here with us today. It was truly a remarkable day. It kind of flew by, but it was really a treat to be with you. And I hope you enjoyed the sessions, and please know they've all been recorded and will be available in the next week or two on our website and on our YouTube channel. So thank you for joining the summit today. And thanks again, John, for being with us. Everyone have a great rest of your weekend. Bye-bye.